Welcome to another video tutorial. In this video, I will introduce you to a new database tool which is called NMR database. And actually this NMR database is also a web-based tool, as we have previously explored different web-based tool. And actually this NMR web-based tool is far more powerful than we have explored all the previously web-based tools. And as here you can see, you can access to this web-based tool by entering our URL, that is bioinforw.com. and if you cannot access to this website as it is in Chinese, so you can simply write bioinforw.com, and once you will click, and there will be many options, so you can directly go to the login page first. And as this web-based tool is in Chinese, so you can use the translation feature. I mean, like this, you can simply go with right click and translate to English, so all the Chinese content will be translated automatically to English. So first of all, you need to register, and registration is totally free, and here are its different version, and you can go to the simple data mining. So once you will click on this, so you can see here we have different options, like for analyzing data, for processing data, then we have also file management. If you are using free version, so I believe there would be no cloud space. We can click on analyze, and after that you can see we have different categories for different type of bioinformatic analysis. So first of all, we can go to different data tool chain and ngplot, and as we have explored in sorplot and different other web-based tool, all the basic setup of those statistical plots is almost same. I mean, if you want to draw anyone, you can simply upload your data and choose different options for your final output, and we can draw our final drawing. And we will test this feature later on in a separate video, but in this video we will only explore the different options. So we can go back to NewMer, and we can go to data mining, and we can go to analyze, and first we can start looking at the collection and the statistics. And as we know, for statistics we usually need like student t-test, we have correspondence analysis, we have multi-dimensional scaling test, we have time series decomposition, we have ADF test, then we have regression and t-test, and even simple ANOVA we have. You can see we have repeated Meyer ANOVA, one-way ANOVA, covariance, and I believe, I mean, all those tests which we usually require in our thesis writing or manuscript submission, all those statistical analyses are available here. And in the comparison, you can see we have MAFT comparison, then we have BOUT comparison, then we have SOAP2, HiSET2 alignment, then we have HiSET2 index creation, RNA sequencing, then HMM scan. And here we have one option for notes. I mean, we can go for gene annotation using eggnog mapper, and we have metagenomic species annotation. And for DNA structure, you can see we have different option for gene prediction, RNA prediction, and we have also option to extract the glimmer sequences as well. And for evolution, we have IQ tree constructions, and for assembly we have Trinity assembly, we have de novo assembly for eukaryotic transcriptomes, and then we have string tie and gap closer. I mean, I believe this would be the really comprehensive tool, and we will explore one by one its different option in later on videos. And here we have different option for quality controls. You can see we have fast P quality control, then we have fast QC quality control, then we have ribosomal RNA contamination detection, and for SNP and indels we have QQ plot for GWAS, and we have Manhattan map, then even we have the complete GWAS analysis, then we have Mendelian randomization, and then we have form processing. This would be a little simple, so we can move to the sequence processing. And as you can see, sometimes it is tricky to extract the faster sequences, and here we can according to GTF file, and here we can according to the column list, and here we can extract the meta gene predicted sequences and FASTA sequences sorting. And for quantitative analysis, you can see we have Salmon index and Callisto build index, and we have chromosome coverage analysis, sequencing depth calculation. And for machine learning, I think this would be interesting and really new for a few people. And here you can see we have clustering, and we have correlation, we have confidence ellipse in Python, then we have random forest model training, then we have boost meta algorithm training, and we have PCA clustering, k-mean analysis, and many more. And if we can see in RNA, you can see here we have RPFAM, and we have coding region, linker RNA, and coding region prediction. Then we have different options for format conversions, and what we have more in price 2, here we have different single cell sequencing. I mean, we can rename group, we can use different gene list, then we have scan py, then we have Scivolo, and we have monocular harmony. And for spatial transcriptome, we have spatial visualization, then we have spatial scampi, then we have spatial scRNA lab, and we have options to merge the sample and many more. So if you want to try this software, so we will try one by one different tools in our later video. If you have any question related to the use of this NewMer tool, please let me know in the comment section. Thank you for watching. Bye bye.